Yes, today we are going to start with a very new topic, that is a very important creature and very interesting creature called virus. Why we are calling it a very interesting creature? Because it has a dual nature. Dual nature. What do you mean by dual nature? Dual nature means it is having the characteristics of both non-living as well as living organisms. So that is why we are calling it a very interesting creature, because it is having non-living characters as well as living characters. So we are not explaining it as an organism or a complete organism. We are explaining it as a particle. So a virus is a particle, not a complete organism, comprising of a package of genetic material and protein. Now here the genetic material can be the DNA or the RNA. And the protein. So a virus is a particle comprising of a package of genetic material and protein. Genetic material can be DNA or RNA. So the virus is not a complete organism, it is a particle. Now, the discovery of this special creature leads to back the scientist by a Russian botanist, Ivanovsky. That botanist or the scientist Ivanovsky discovered this special creature called the virus, which have the characteristic of both non-living as well as living organisms. Now, the characteristics, that is the non-living characteristics. Why we are calling it a particle? Because of this non-living characteristics. Number one, non-living character is what? That is, it do not possess cell membrane. Next, it lacks respiration. Next characteristic on the basis of which it is termed as non-living is it is devoid of metabolism. That is, it do not possess any cell membrane or cell wall, it lacks respiration, and it is devoid of metabolism, on the basis of which we can consider it as a non-living substance. That is why it is called as a particle. Now we are considering its living characters. Why this can be characterized or considered as living? On the basis of that it forms its replica. So what do you mean by replica? It reproduces or it forms its own copy. That is why it is called a living characteristic. Next thing is that it possesses genetic genetic material, that is DNA or RNA. DNA or RNA. Next thing is, it can cause disease. It can also cause diseases just like bacteria or the fungus. That is why it is considered as the living character of the organism. So it is having non-living as well as living characteristics. So it is a very interesting creature discovered by the scientist Ivanovsky, a Russian botanist. Now we are going to classify them on the organism to which they infect. So, characterization or category. The characterization or the category of the viruses. So, first of all, animal viruses. Next one is plant viruses. And third one is bacterial viruses. Third one is bacterial viruses. Animal viruses are also called zoophagini. Zoo is animal, and plant viruses are called phytophagini, and bacterial viruses are simply considered as phagini. So zoophagini, those viruses which causes effect on animals, phytophagini on plants, and phagini for bacteria. That is those viruses which infect bacteria, and these are especially considered as bacteriophages. Now, among all these, we are most considered with the bacteriophage virus. Now, coming on the next, so the characterization, the categorization of the virus is based on the organism which it will infect. Now, coming on the point, how they form its replica. They do not possess any type of cell-like structure. They do not have cell organic, whether it is of prokaryote or eukaryotic. But in spite of that, it performs some living characteristics. That is, those characteristics which are found in living organism. That is, it forms its own copy. That is replica. Now we will see how it will form its replica. So, reproduction. So, it performs or it forms its replica with the help of Lytic Cycle and the Lysogenic Cycle. Lytic Cycle and the Lysogenic Cycle. Now first we are going to deal with the Lytic Cycle. What is this cycle means? Lytic Cycle. Lytic means lysis. Lysis means to break. So what happens in this? The virus enters the cell or the organism or the cell of the organism and takes over the machinery, takes over the genetic machinery of the organism and multiply manifolds. Manifold means in multiples of the original number. The virus enters the cell and takes over the genetic machinery and forms the new viruses or kid viruses and causes the breakage of the cell, and this is called the Lytic cycle. When it will cause the breakage of the cell, it is called Lytic cycle. It can reproduce, or it can enter the cell and reproduces or makes its own copy with the help of the machinery of the genetic machinery of the host, not itself, because it do not possess any type of machinery for the reproduction. And next is the Lysogenic cycle. Next is the Lysogenic cycle. Here, virus is very smart enough. In the earlier chapters we have studied, we have already discussed that it is an interesting creature. Why? Because it is performing a different type of biotechnological application from its beginning. So in Lysogenic cycle, what it does, the virus cuts the genetic material of the host and ligates its own genetic material with the host genetic material. That is, it is forming a combination of the genetic material with the host material. Say for example, this is the genetic material of the host, and it cleaves the genetic material of the host and attaches its own genetic material, so that when the cell divides, it also gets divided. That is, it forms a sort of recombinant DNA. Recombinant genetic material or recombinant DNA, if the virus is of DNA genetic material. So when the cell divides, it will also divide and duplicates and increases its number, and in different circumstances it will cause the breakage of the cycle and breakage of the cell and releases out. So what is the basic difference between the lytic and the lysogenic cycle is what? That in lytic cycle it do not causing the formation of recombinant DNA in the lytic cycle, but in lysogenic cycle it forms a recombinant DNA or the combination of the DNA. So it increases its number and releases the kid viruses or the new viruses in the new cells to infect. So this is how it causes the increase in number of the virus in spite of being inactive outside the host. It reproduces, it increases its number and causes harm to the animals, humans as well as plants. And in fact, a very notorious organism, the bacteria, is also infected by the viruses. Now we have very good interest in the Bacteriophage virus. In the Bacteriophage virus, because this can be of utmost importance for us, because the bacteria causing different types of diseases, and if we can use the Bacteriophage virus for destroying the bacteria, then we can cure or we can prevent many of the infections or many of the population related infects. So bacteriophage virus. Now we are going to draw a general diagram of the bacteriophage virus. This is a bacteriophage virus. It is having a head, a collar, and tail fibers. Head, collar, tail and tail fibers, and having the genetic material as double-stranded DNA. It is having double-stranded DNA, bacteriophage virus. So this virus is actually responsible for the neatness of the river Ganges water, because it destroys the bacteria in the water and causes prevention of the water from pollution. And this is very important creature because it helps in the decipherance of to decipher the nature of the genetic material and to decide whether the genetic material is DNA or RNA or not the carbohydrate, not the protein, not the lipid. It is because of this bacteriophage virus. This bacteriophage virus, and you can say how it is important, how it is going to decipher or how it helps the process of deciphering that the DNA is the genetic material and not the carbohydrate or protein or anyone else by Hershey and Chase experiment. Now we are going to see Hershey and Chase experiment. Hershey and Chase experiment. This experiment was conducted by Hershey and Chase to prove that DNA is the genetic material. DNA is the genetic material. So the procedure adopted was number one, what he does, he has taken the bacteriophage virus and tagged it with the radioactive phosphorus. This is radioactive phosphorus. So as a result, the phosphorus gets incorporated in the genetic material, radioactive phosphorus, because phosphate is present in the genetic material. In the next, what he does is he tagged the radioactive sulfur. Tag the radioactive sulfur with the bacteriophage virus. Now what he found, that the protein code is tagged with the radioactive sulfur and not the radioactive phosphorus. So in the next step, what he does, they allowed the radioactive sulfur tagged bacteriophage viruses to infect Escherichia coli, that is the bacteria found in our colon. So he does not found any radioactive genetic material in the E. coli. But when it here they allowed radioactive phosphorus tagged bacteriophage to infect Escherichia coli. Then they found radioactive DNA tagged with radioactive phosphorus. So this proves that it is the DNA and not the carbohydrate or protein or something else which is going to transfer from one organism to the next organism, and DNA is the genetic material. So this guy, this bacteriophage is very much important because of the disappearance or the discovery of the genetic material is DNA and not the anything else. So bacteriophage is having the main role in the genetical problems as well as genetics whole. So bacteriophages can be used for our protection, for our welfare, human welfare as well as our cattle welfare as well as our plant welfare or the crop improvement. So this bacteriophage. So now up till now we have seen the bacteriophage is very important for us and for the cattle yield as well as plant yield. Now this bacteriophage virus is as well important in controlling water blooms or algal blooms. Algal blooms that is caused by blue-green algae or the member of Cyanophyceae That is why this type is known as cyanophages. And those causing harm to the fungi, they are known as mycophages. And those causing the different types of alga, then they are known as phycophages. This is causing the harm to the blue-green algae, this is to the fungi, and this is to the different types of algae which can harmful for the aquatic life as well as for the humans. Now the topic virus cannot be completed without a notorious virus, without the explanation of a notorious virus, that is HIV, a well-known, a deadly virus, that is human immune virus. Human immune virus. This virus is responsible for most of the deaths in the world and most appropriately in the sub-Saharan region of Africa, because it causes effect on the resistance power of the body, that is, it decreases the resistance power of the body. But one thing should be in mind, that the presence of the HIV in the body is considered as positive, but the presence of HIV in the body, that is, the person may be HIV positive, but it may or may not have the AIDS. So AIDS, it is not definite that the presence of HIV positive, the HIV positive does not mean the person is having the AIDS. AIDS is the last stage of having the HIV. First of all, it counteracts or it creates AIDS related complexes. Then the last stage is AIDS. So a AIDS patient is not dying of the AIDS. It is dying because of different types of opportunistic infections. What do you mean by opportunistic infections? Opportunistic infections. Opportunistic infections means HIV is going to reduce the immune power, that is resistance power of the body, and this causes the effect of different types of microbes or the pathogens, which is going to have the advantage of the decrease in the resistance power of the body and can infect the body and causes the diseases. So a person counteracting with the AIDS is dying because of different types of infections, and that are known as opportunistic infections, because they are having the pathogens of different types of diseases is having the opportunity or the advantage to enter in the field of your body. So it reduces the immune system and different types of pathogen is going to attack and causes different types of diseases. So now let's have a look on the general structure of the HIV. So I am drawing the general diagram. As we know that a virus is a nucleoprotein. A virus is a nucleoprotein. That is, it is having a nucleic acid, that is DNA or RNA, and protein coat. So we have these are RNAs and these are proteins. General structure and indicative structure. GP120 and this is P17, and these are single stranded RNA. Single stranded RNA. Single stranded RNA. These are not double stranded. These are single stranded RNA, but two copies of single stranded RNA. And these are reverse transcriptase. These are reverse transcriptase enzymes, helps in the formation of DNA from RNA. So all those viruses having single stranded RNA comes under a group retrovirus. Retrovirus. So retroviruses are those which have single stranded RNA, and it is a single stranded RNA having virus. So the RNA containing, as we have already said, that the DNA or the RNA can be the genetic material of the virus. But RNA viruses are much more lethal than DNA viruses. Why? Why? Because RNA is more prone to mutation. Mutation means the sudden change in the genetic material. So this HIV is much more lethal because its genetic material is RNA. Because it is much more labile, it is not stable as comparison to the DNA. So it is having an extra hydroxyl group at the second carbon in the RNA. That is why it is more labile and prone to mutation. So when you are making the medicine for staking or obstructing any of the stages, it is going to change. It is going to change itself and that medicine becomes waste. That is why this becomes widespread and it is hard to kill all the viruses in the body. But there are some retroviral or antiretroviral medicines like AZT, azithromycin. But what we are going to see that how it is going to infect the cell or infect the body of the organism. What it does when it enters the body, here I must recall that the body having the HIV, that is HIV positive, does not mean that the person is having the AIDS. So it becomes dormant after entering the body of the organism. Dormant means it becomes waste, but it becomes active after 5 or 10 years depending on the resistance power of the body. So the HIV is going to affect T lymphocytes. Is going to affect the T lymphocytes. You must be aware of the T lymphocytes and B lymphocytes. This T lymphocyte as well as B lymphocytes both are going to attack on the intruders or the foreign agents which can cause the disease. So these T lymphocytes are of 4 types: TH, TK, TS and TM. That is killer cells, helper cells, suppressor cells and memory cells. Killer cells is going to kill directly by attaching with the pathogen by producing interleukins or perforins. But helper T cell performs dual function. It activates B cells, that is B lymphocytes, and it also produces interleukins to kill the pathogen. So the HIV is intelligent enough to attack the TH. It does not destroy, it does not attack the killer T cell. It destroys the helper T cell. So as a result, there is double attack on the body that it activates, because helper T cell activates B cell and interleukin. So there will be double attack and deactivation of the B cells and interleukins. As a result, it can act on the body and causes the cell to form new HIV with the help of reverse transcriptase enzyme, that is formation of DNA from RNA. The formation of DNA from RNA. So this process is known as reverse transcription. That is, but the usual central dogma of molecular genetics is DNA to RNA and RNA to protein. But this HIV is going to form DNA from RNA. That is reverse transcription, or it is known as teminism. Teminism or reverse transcription. With the help of enzyme reverse transcriptase. Reverse transcriptase. So with the help of reverse transcription and with the help of machinery of the cell, it forms new viruses with the help of reverse transcription and reverse transcriptase enzyme, and causes the most lethal, up till now, the most lethal problem is AIDS. So HIV can be controlled by precaution. So the say that the precaution is better than cure is very true in this regard. So prevention is better than cure. So viruses. So as we have seen that viruses are nucleoproteins. Nucleoproteins. That is, it is having the nucleic acid and proteins. Nucleic acid and the protein. But some of the creatures, or you can say the siblings of this virus, which also causes different types of diseases, are viroids and prions. Viroids and prions. These viroids have only nucleic acid and no protein coat. That is simply, it is a infectious RNA particle. So a nucleic acid can also be infectious. Now the prions, this is only protein particles, and we can say it is infectious proteins. Infectious proteins and no nucleic acid, and it can cause a very lethal, or you can say the interesting disease called Kuru. This is found in African tribes which are cannibals. Cannibals means which eats or which ingest or which consume its own species. So they are consuming their own species. So this type of diseases are more prevalent in that tribes, that is African tribes of sub-Saharan region. So viruses, viroids and prions, these creatures are having a very different and very unique effect on the life of the human being as well as animals as well as plants. So they should be studied properly to curb or to increase the production of the crops as well as for the benefit of the human beings. Thank you.